Hello everyone, and thank you for taking interest in my poster. My project focuses on the impact of copper complexes on P-glycoprotein expression and function at the blood-brain barrier. PGP is often referred to as the gatekeeper to the brain, as it keeps a large range of molecules from entering. This is great in terms of protection, but becomes a hindrance when trying to develop drugs to be delivered to the brain. PGP is also involved in the clearance of endogenous molecules such as amyloid beta, and studies have shown that PGP expression and function is reduced in people with Alzheimer's disease, where an excess of amyloid beta is thought to be a major pathological hallmark. Interestingly, in AD, there is a decrease in intracellular copper within cells of the central nervous system, and copper is vital for maintaining the integrity and health of cells within the brain. Preliminary data from our lab suggests an association between levels of copper and PGP expression. So one of the aims of this project was to deliver exogenous copper into human brain endothelial cells using copper GTSM, which is a complex that releases copper within cells. And copper ATSM was used as a negative control that only releases copper under hypoxic conditions. Protein expression was assessed by Western blot analysis, and what we found was a complete surprise. The negative control, copper ATSM, increased the abundance of PGP following all treatments. But what was even more unexpected was that copper GTSM, the giver of copper, had the opposite effect and diminished PGP expression at the longer 48-hour treatment. PGP function was assessed by measuring the accumulation of known fluorescent PGP substrate, rhodamine 123. As expected, there was decreased accumulation of rhodamine following treatment with copper ATSM, indicating an enhanced efflux function, whereas copper GTSM showed a reduced amount of efflux. To assess whether the altered expression of PGP protein was via a transcriptional mechanism, PCR was used to quantify PGP mRNA levels over a series of time points. And indeed, it confirmed that protein upregulation was induced by a gene transcriptional activation or inhibition by copper GTSM. Both copper ATSM and GTSM showed a concentration-dependent elevation of intracellular copper levels, but whether the copper detected by ICP-MS was free bioavailable copper or complex-bound copper remains to be determined. But nevertheless, the two compounds are indeed entering into the brain endothelial cells to change the dynamics of this key efflux transporter at the blood-brain barrier. The results to date may suggest that PGP expression is mediated by a copper-independent mechanism, and it may be the complex backbone structure itself that elicits the responses seen in these cells. And further investigation is currently underway to confirm if this is the case. Thank you for listening.